Hi everyone, I'm Fabrizia Fusetti, Scientific Director Bioanalysis at QPS. It is a pleasure for us to present one of our latest publications on the development and validation of a hybrid LCMS method for the simultaneous determination of Trastuzumab and Pertuzmab in clinical study samples. The work is a result of a collaboration with the Department of Oncology of the Amsterdam University Medical Center, and I'm pleased to introduce our closest collaborator, Sandor Schokker. Welcome, Sandor, and thank you for accepting to contribute to this presentation today.
Hi everyone, I'm Sandor Schokker from the Amsterdam University Medical Center, and I would like to thank QPS for this wonderful collaboration and this opportunity to present our results.
Well, Sandor, it has really been fun to work together with you during the past years, and I would like you to start now by introducing the project, and then I will continue to describe the methods, and then we'll guide you all through this publication.
All immunoglobulins are almost identical within a species, and sometimes that could be an advantage from a bioanalytical point of view. Trastuzumab and Pertuzmab are humanized therapeutic antibodies targeting the human epidermal growth factor receptor 2, or also known as HER2, which is highly expressed on the surface of various types of cancer. They have a complementarity mechanism of action because Trastuzumab effectively blocks the HER2 receptor and subsequent intracellular growth signal transduction, but does not prevent heterodimerization with HER3, which is one of the Trastuzumab resistance mechanisms. Pertuzmab blocks heterodimerization effectively and thereby overcomes this resistance mechanism and has a complementary mechanism of action, which makes this combination a good combination in clinical practice. And the combination is being used more and more. Sensitive ELISA assays are available for both monoclonal antibodies and commercially available. However, PK profiling in support of experimental trials where the two antibodies are co-administered would require repeated sample analysis with considerable increase of cost and use of sample volume.
Well, that's why we decided that mass spectrometry would be a very good alternative for ELISA for this project. And then one essential critical point, was that the two molecules could not be distinguished by the same within the same assay, and that's why we decided to use mass spectrometry. In fact, to reduce immunogenicity, humanized therapeutic antibodies are built by CDR grafting, which results in molecules that will be largely identical to human IgGs, except for the antigen binding site on the variable chain. Some regions display 100% sequence identity, and other around the antigen binding site or the CDR will be completely or slightly different. So this is exactly the case for Trastuzumab and Pertuzmab. These two antibodies are almost exactly identical, except for the region that on this slide are highlighted in orange and red, that are different because they are responsible for the recognition of the antigen and the different parts of the receptor. A hybrid mass spectrometry assay adds the additional level of selectivity that is needed to distinguish these two molecules that are almost identical, and it works even when molecular differences are very small. On top of that, the multiplexing ability of hybrid methods will allow the quantification of more antibodies in one single assay, which was of great advantage in this case. But differently than for ligand binding assay that normally measure molecular interaction, what characterizes the hybrid LCMS method is that the target molecule is extracted or enriched from the sample by an affinity purification step. The affinity purification is mostly based on magnetic beams and is followed by proteolysis that reduces the protein in peptides. The aim of the sample preparation is to increase recovery and reduce complexity and background interference to achieve maximum sensitivity. The sensitivity will result from optimization of all the steps. And in general, the more specific the affinity purification, the lower the background, the higher the sensitivity of the assay. Essential for minimizing technical variation during sample preparation is the addition of an internal standard that should display similar or identical physical and chemical features of the analyte, which allows monitoring of the full sample preparation workflow. In this way, LCMS will produce a measurable response for the surrogate peptide that is obtained after proteolysis. And the assumption is that the concentration of the surrogate peptide will be stoichiometrically equivalent to the concentration of the target protein. For this specific project, we use a non-selective affinity purification with protein A. We use a very small sample volume of 10 microliter. In our hands is a small sample volume often better in terms of reducing the interference of the background. And in this case, it was advantageous because we didn't have to use a large amount of sample. During the method development, it became clear that although none of the peptides contain cysteine residues and the CDR region is exposed on the surface of the antibody and therefore readily accessible to proteolysis, the addition of a reduction and alkylation step contributed to an increased and consistent recovery of the tryptic peptides. The amount of trypsin, 20 microgram per samples, and the incubation time, 60 minutes, and temperature, 37 degrees, were optimized for sufficient recovery of peptides and allowed to obtain a reproducible and time-effective sample preparation without introducing too much stress in the sample. The peptides that were chosen for selectivity and reduce interference were one peptide for Trastuzumab and one peptide for Pertuzmab, and one peptide for the LC-LCIMS that was used as internal standard. The internal standard peptides was not chosen because it was a true stable version of any of the other surrogate peptides, but it was chosen because its retention time was closer to one of the surrogate peptides. And in this slide, you can see that the chromatogram, and then you can see that the peak produced by LC-LCIMS is almost identical, or it has almost identical retention time as the peak from Pertuzmab. Just before I mentioned that we were aiming at reducing stress during sample preparation, and that has also a specific reason. The CDR peptide is frequently used as a surrogate peptide for Trastuzumab quantification. And in this assay, it was critical for discriminating between the two molecules. But it contains an asparagine residue that is followed by a glycine, which then constituted an G motif, indicating that the site is susceptible to deamidation. Deamidation of asparagine is a common post-translational modification that can occur both in vitro and in vivo, and it can affect the functionality of Trastuzumab. It is known that in vivo, it is mostly causing conformational change and is probably affecting the binding to the HER2 receptor, compromising the functionality of the therapeutic antibody. As demonstrated by the data that was published in this article, of which I'm showing some of the graphs, and the work was from the Department of Analytical Biochemistry of the University of Groningen. The temperature and pH stress can have a great impact on the degree of the deamidation of this spot. And as we were aware of this feature, we care to reduce the molecular stress to the minimum during sample preparation, which takes in total only 4 hours. Here I'm showing a summary of the results from the validation with a specific focus on the precision and accuracy data. The results from the validation of the method are fully described in the article that we published. And I will only now assure you that the assay passed all items. And we added also a reciprocal interference test to assess the impact on the recovery, and that also showed that that was could be kept to the minimum. The method shows very good accuracy and precision, and it is very selective. The parameters were set to comply with the regulatory recommendation for large molecules, and you can see that the statistics are very good. And even better for Pertuzmab, and we think that that might have to do with the fact that the internal standard could be better, could be more representative for this peptide, for the surrogate peptide, than for the Trastuzumab. These were the result of the validation. However, after having followed the clinical study, we had more batches and thus more statistics to, or let's say more results to build a better statistical performance of the assay. And in this slide, you can see that both the precision and accuracy of the assay are becoming very good across many batches, having parameters below 10%. Which says really a lot about the accuracy and the reliability of the data that was measured in this sample. And then after this, I would like to give the floor to Sandor that will tell us more about how the assay was used in the clinical trial and give us share also some results on this very important clinical evaluation work. Go ahead, Sandor, please.
Thank you, Fabrizia, for also introducing the TRAP trial, which was a clinical trial in the Amsterdam University Medical Centers and other participating centers in the Netherlands, which enrolled 40 esophageal adenocarcinoma patients that were treated with Trastuzumab and Pertuzmab on top of conventional chemoradiation. The Trastuzumab was dosed weekly in the first weeks, and after that every 3 weeks. The Pertuzmab was dosed in a fixed dose every 3 weeks. The method that Fabrizia just described was used for to assess the exposure-response relationship and also the relation between pharmacokinetics and toxicity. In short, the results of the TRAP trial, the progression-free survival at 3 years was 57%, and the overall survival was 71%, with a higher percentage of HER2-positive patients with a +3 overexpression being alive at the 3-year time point. In total, we had 670 study samples where we performed pharmacokinetic analysis, and there was no correlation with survival, pathological response, or with toxicity. On the next slide, we see all the individual data points for the 40 patients. And Here you can see that the results are consistent with the dosing schedule, and on the next slide we can even see better that all the results are within the assay range with a C- and C max that are well within the assay range, and all the study subjects reached a minimum C- of 20 micrograms per milliliter. And on the next slide, I wanted to discuss that since combinations like Trastuzumab and Pertuzmab are increasingly used in clinical practice, also with other monoclonal antibodies, there will be a higher need for accurate multiplex pharmacokinetic assays. And for multiplexing pharmacokinetic assays, and in general, when a high degree of selectivity is needed, hybrid LCMS-MS assays, even with some limitations in sensitivity and costs, represent the only alternative to the traditional ligand binding assay formats. This hybrid assay can determine nanomolar concentrations of Trastuzumab and Pertuzmab. It's robust, fit for reliable determination of PK profiling of the currently administered doses of antibody therapeutics in clinical studies, also throughout a long period of time. The method, validated according to the current FDA and EMA guidelines for large molecule bioanalysis, is accurate and selective for the simultaneous determination of Trastuzumab and Pertuzmab in clinical samples. It has a medium-high throughput and overcomes the limitation of ligand binding assays that are challenged by MAPs targeting the same receptor. As the sample preparation is generic, it can easily be adapted to multiplex quantifications of other MAP combinations in non-clinical and in clinical samples.
Well, Sandor, thank you very much for contributing to this presentation. And on behalf of QPS, I would also like to extend my thanks to Professor Hanneke van Laarhoven, which was supervising the research in a group at the Cancer Center in Amsterdam. And from the Netherlands QPS team, I would like to thank the large molecule mass spectrometry group that has developed the assay and also follow and manage the bioanalysis part for the clinical study. Thank you very much, Sandor, for being here with me today.
Thank you so much for the invitation.
Well, the validated assay that we presented today is described in this article published in Meds in August 2020. The assay is available for PK determination of Trastuzumab and/or Pertuzmab in clinical studies. And if you are interested or you would like to know more about our hybrid mass spectrometry bioanalytical platform, please get in touch at info@qps.com. Thank you very much for listening.